Hello friends, it's Professor Jia Gowda once again before you to teach that points that are neglected and not clarified during teaching and learning process of biology. Already in my previous episodes I have clarified some 2-3 points of flower and oxygen's utility and all that. Now in this episode I am going to explain you how the plant cell must be drawn, how the animal cell must be drawn. Some draw very beautifully, of course. And nowadays, the number of explanations about cell structure and all that are coming in the social media, like YouTube, Facebook, and all that. But there are, I'm observing number of defective clarifications there. For example, in the one that I'm going to now show you here, these two videos. These two videos I observed and heard the clarifications. The lady that she is dealing with cell structure in one episode, she told that animal cells are oval, plant cells are hexagonal. Both points according to me are wrong. I am not inserting that teacher, please excuse me, but the reality is animal cells need not be oval, plant cells need not be hexagonal. But of course, in the plant cells walls, there are corners coming while we are drawing. Whereas in the animal cell, corners are not coming. Why it is? It's not clarified properly. And the word oval is used. No, oval means egg shape. The person who is explaining the plant animal cell as oval, actually diagram drawn is oblong. It's not oval either. So when we are using some words, they must tally with the clarifications. Let it be. But the thing is, see, animal cell and plant cell, how we have to draw, why we have to draw like that only is a very important aspect. I'll show you now. See, animal cell and plant cells are drawn in this way. This is animal cell. This is animal cell drawn like this, with say this outer cover with only 2 lines as outer cover. 2 lines with outer cover here, like this, 2 lines. Whereas in plant cell, see, in plant cell we draw like this. Here we are drawing 3 lines. See, 1, 2, 3 like this. Outer cover, 3 lines. Why this is? It's not explained. First of all, these figures are ultra structures. But nobody has used this word ultra structure. Means what? Students must know. Ultra structure means as seen under electron microscope is the meaning. So the diagrams that I am showing you now, these are ultra structures. Ultra structure means as seen under electron microscope, but we are not seeing the cells in the common Leeuwenhoek's microscope like this. So ultra structure, when we see like this, ultra structure means in terms of measurement is in terms of microns and angstrom units. What is that micron and angstrom unit means? Here I am giving you the details. You can see 1,000th of mm equal to 1 micron, 1 mu, otherwise read as. Now 1,000th of mu or micron equal to 1 nanomicron or 1 micromicron, like that. Then 1/10th of micromicron, micro mu equal to 1 angstrom unit. So these are the measurements commonly under electron microscope we do in case of biological observations. Of course, see, these ultra structures, when we are drawing outside here, this 3-layer plant cell, what is this inner is the cell wall of this cell. this cell, original one cell's inner cell wall. This is cell wall of this cell, and this is cell wall of this neighboring cell. Outside here there is a cell, outside here there is a cell, outside here there is a cell, like that. Here 6 corners we have made, means 6 neighboring cells are there. Their walls they are primary walls, and in between these two primary walls, this primary wall of this cell, this primary wall of this cell, they are separated by middle lamella. This is middle lamella. See, actually when we draw plant cell, why we have to draw outside cells, neighboring cells? Because neighboring cells also we draw, because in between the neighboring cell and this main cell there is middle lamella. This is middle lamella, middle lamella, this is middle lamella. This middle lamella is contributed by both the cells, this main cell as well as this neighboring cell. That's why middle lamella belongs to this also, this also. And if we don't draw middle lamella, it means that the one cell we are drawing, its one portion we are not drawing is the meaning. But if we don't draw this outside cell, then it means that this middle lamella belongs to fully to this cell only. This cell only. It's not so. It belongs to both the cells. That's why when we have to draw middle lamella, and if we draw middle lamella, its contributor neighboring cell also must be drawn. That's why we draw it like this. So this is not told by any clarifier or any teacher I have seen so far. That's why it is essential. Students must know this. And students are also blindly drawing this without inquiring, without asking anybody why these 3 walls only, 3 lines only, that nobody is asking. Okay, let it be. Now see, one more diagram I am showing. I am showing a plant cell here. With the inner one, this is the primary wall of this cell. This is secondary wall of this cell. Primary wall. Primary wall is here. This is primary wall. This is primary wall. That is, what is that? Violet colored. This primary wall of this cell, and this is secondary wall. This is membrane, plasma membrane of this cell. Okay. Likewise, this green one is middle lamella. After middle lamella again, there is a primary wall of this neighboring cell, violet in color, primary wall of this neighboring cell. Then secondary wall of this neighboring cell. Like that, see, secondary wall, of course, I have shown with orange color in both the sides. Secondary wall of this cell, secondary wall of this cell, I have shown with orange color. Secondary wall is not there in this diagram. I have not shown it in this diagram. Because secondary wall presence is not compulsion. It may be there or may not be there. Secondary walls are there only in some cells like sclerites, stone-like cells. Stone cells actually they are. Sclerites, one type of modified sclerenchyma cells. They have secondary wall. And if secondary wall is there, of course it has to be shown. But secondary wall may not be there. That's why common in an ideal diagram, normal diagram like this, we are not showing the secondary wall. Primary wall only we show. Secondary wall, if present, sometimes it is so thick that this entire lumen of the cell, this entire lumen is completely filled by secondary wall only. And the entire protoplasm, this nucleus of course, the nucleus and the cytoplasm, both together protoplasm, both will disappear only, and cell is a dead cell at maturity. So entire cell is filled with secondary wall. It is like, that's why it's a piece of stone only. That's why it's called stone cell, sclerite. But secondary wall, if necessary, then only it is there. In all plant cells it is not there. That's why commonly it is not drawn. This point is not clarified. And ultrastructure word is not used in the explanations. Of course, it may be there in the textbooks as a headline for these figures, but teachers are not using it. That is wonder. Must use, and they must clarify why ultrastructure is called. They must tell. That's not told. That's why I feel like touching these points which are not tackled at all. So then, some say that plant cell has a large vacuole. Of course, it's right. Plant cell has a large vacuole, but at the beginning, when the plant cell is small one, the vacuole is not there at the beginning. Slowly it goes on growing. That's why it may be of any size. But in a mature cell, it's of course large one, largest one in the cell among all the organelles. And that's why nucleus is pushed to the side like this. It's not in the center of the plant cell. And in animal cell now, nucleus we commonly draw in the middle because vacuoles are very, very small or they are absent also. That is. Then endoplasmic reticulum, Golgi bodies, mitochondria, all these things we draw. But how many mitochondria? See, for example, this is a mitochondrion. I'll show you. This is a mitochondrion. This is a mitochondrion. And this is a, see, this is a mitochondrion. In animal cell, mitochondria are the mitochondria. There, lysosome is there in animal cell. Lysosome we draw. And mitochondria, how many mitochondria have to be drawn? So in animal cell, mitochondria we draw. In plastids we are not drawing. But in plant cell, see, this is mitochondrion. And this is a plastid, chloroplast. This is chloroplast. So there are mitochondria and also chloroplasts in plant cells. And how many mitochondria we have to draw in a cell? Mitochondria number is not definite. It varies from cell to cell. They are more in active cells, means actively working cells where more energy is to be released. They are more, especially in cells of people like, for example, say, in cells of people like Salman Khan, you can say, Salman Khan, and that's bodybuilder. And Hima Das, the athlete, national athlete from eastern state, Assam, I think, yes, from Assam area. Hima Das, athlete, powerful runner. So in such people, their muscle cells are releasing more energy because they want more energy, and when they are exercising and performing their profession that they are meant for, they are doing actually, their more energy is released. That's why mitochondria are more in them. But in other cells, mitochondria are less. In some cells of our body only, mitochondria may not be there also. That's why there is no definite number. But commonly we draw 2-3 mitochondria. And plastids also, there is chloroplast or tomoplast or leucoplast. So when plant and animal cell differences are told, plant cells have plastids, they say. Animal cells don't have them, they say. There are plant cells not having plastids also. For example, chloroplasts are absent in underground parts of the plant, like roots or any underground portion storing food. In such things, chloroplasts with non-green parts of the plant, I mean, like roots especially, there are no chloroplasts at all. Plant cells only there also. That's why these things are not elaborately explained. So in explanation, the general idea they fix in the minds of the students that plant cells always have chloroplasts. No, always they need not have. Only photosynthesizing cells have. Others don't have. Like that, these things like presence or absence of plastids, quantity of them, quantity of mitochondria, and the ultrastructure, such words are all neglected. Such neglected biological teaching I want to stress and highlight. That's why there are so many things to be in ultrastructure. In explanation of that, micron, angstrom units are also not so much clarified. The words are not used also by a number of students. And if you question them many times, many bright students are also silent on these things. But everything in detail, if known, then only one can compete for competitive examinations. That's why the points that I am touching now are essential points. One has to realize that they must be well prepared and well known, well informed in these things so that they can be successful in competitive examinations. So here in plant cell, plasma membrane, where is plasma membrane? I told that this is the primary wall, this is the primary wall of this cell. If it is primary wall, then inside to this plasma membrane must be there. We are not drawing it because plasma membrane is the first cover for protoplasm. We are not drawing it in the plant cell because, why we are not drawing? Because the plasma membrane is adpressed to the cell wall, adhering to the cell wall. So it's not visible clearly like this. That's why it is not drawn. And in animal cell, it is drawn with two layers. Why? Because see, plasma membrane is a double structure. Fluid mosaic model you must have studied. Fluid mosaic model of plasma membrane you must have studied. I will show you that one now here. See this fluid mosaic model of plasma membrane. I will show you 1 minute. See this please. The fluid mosaic model that you are now seeing is actually a double layer. This is one layer, this is another layer of lipoproteins. So these proteins, lipids, proteins and lipids. Like that, see, this is one layer of lipoprotein, this is another layer of lipoprotein. Since plasma membrane is made up of, this is also ultrastructure, and it is drawn in that diagrammatic diagram. In this diagrammatic diagram, I mean in this diagram, it is drawn as two lines. Means these two lines are that lipoprotein layers only. So two layers we draw because of that. Okay. And in one more explanation I have seen, lysosomes, one person I was explaining in this social media only, that lysosomes are suicide bags. The phrase correctly he used, but he didn't clarify that why they are called suicide bags. Suicide means self-killing. Now cell has a lysosome, means it is going to be used to kill itself, is the meaning. Of course. Then to kill itself, why it has to die? Why it is keeping it with it to die? Means actually reason is, it is keeping, it has a lysosome has number of lytic enzymes in it. Means they are going to help the cell most of the time. That is, whenever unwanted things are there in the cell, that lysosome will release the enzymes to destroy them actually. But sometimes by mistake it may release lot of enzymes, and that abnormal release of enzymes may kill the cell itself. It may happen sometimes. That's why it is called suicide bag. This point is not clarified. It is a basic thing that is neglected in teaching. Okay, thank you. It's enough. I'll stop it here now. In next video something more I'll explain later.